Okay, we're going to start. Let me see if yeah, there she is. Okay. Well, welcome everybody here at this thesis defense. And I officially use my hammer, as you can see over here, to open this ceremony, this academic ceremony, in which Miss Abril Gijsbers Alejandre will defend her academic thesis entitled Deciphering the Role of Microbacterial Secreted Proteins Through Structural Biology. May I invite you, Ms. Gijsbers Alejandre, to present a summary of your work and the conclusions of your thesis in the next 15 minutes. And I'd like to give you the word.
Thank you so much. I'll start sharing the screen. Everyone can see it? Well, I hope so.
Yes.
Okay, well, dear Pro-Rector, dear members of the Corona, their family, friends, and audience. In the coming 15 minutes, I will shortly describe what I've been working on for almost 6 years, highlighting the relevance for science and our society. The title of my work is Deciphering the Role of Microbacterial Secreted Proteins Through Structural Biology. And although this title might not clarify for many of the audience about my work, I hope that this presentation will. Okay. Proteins are the most sophisticated molecules in nature. And as I try to depict with this video, they are the ones that bring the cell to life. They are like the people within a city, let's put it this way. Almost every biological mechanism involves the action of one or more proteins. And thus anything altering their function will have implications in the cell's performance, no matter the cell type. For a sperm might compromise its fitness, for a bacterium its survival, or for a multicellular organism like us, it could lead to disease. So by understanding their function, what they do and how they do it, we'll have the tools to fix it when there is a problem. Of course, a powerful piece of information is the structure they have. Within the repertoire of techniques that can give you structural information, in our lab, we mainly use electron microscopy in cryogenic conditions, or cryo, due to its power to visualize from entire cells to single molecules. Significant developments in the last decade and its versatility have made this technique one of the most common and thus important techniques to solve protein structures, helping to understand many biological problems and also leaving no question for the Nobel Prize in Chemistry to some of the pioneers in this technique. One obvious example of the problem solved by cryo, or well, at least that we're trying to solve, and that is close to our life now, is the structure of the spike protein of the coronavirus SARS-CoV-2. Without that structure, we wouldn't have the vaccines that we have now, allowing us to have a normal life, or more or less. Motivated on many more cases like this one, our lab focused on another health problem that is as important as SARS, but that we don't really talk about. Tuberculosis is the oldest disease we have on record, and still, every year, approximately 10 million people get ill, from which 1.5 million will die. Before this coronavirus turned our world upside down, tuberculosis was the #1 cause of death from an infectious agent. And the reason is, well, there are many reasons, but one important is that we use a very old vaccine. It just turned 100 years old last year. And it doesn't work in adults. And okay, if you get ill, there are drugs to treat you. They have terrible side effects, making it very difficult for the patient to adhere to a treatment, which in turn contributes to the rise of drug-resistant TB cases, making it more and more difficult to treat and control the situation. So you might be wondering, why don't we just do what we did for the coronavirus, right? To get a new therapy. Well, it is not that simple. because the coronavirus is actually a very simple particle. It has a lipid bilayer like this one, with the spike protein just sticking out, while Mycobacterium tuberculosis, the bacterium responsible for the tuberculosis in humans, has a very complex cell envelope made of many different layers, as you can see here, creating a waxy coat, almost impermeable. And on top of that, there are very few proteins that we know of that are present in these layers and that are important for the virulence of Mycobacteria tuberculosis. So not many options do we have. One possible option was discovered by comparing the genome of the pathogenic strains like Mycobacterium tuberculosis and the genome of the BCG strain, which we use as a vaccine. What scientists like my supervisor Peter Peters noticed was that non-pathogenic strains are destroyed by the immune cells like this macrophage through a process called phagocytosis. But the pathogenic strains escape that fate thanks to a secretion system called ES61, which is absent in the BCG strain. They realized the importance of this secretion system once they added ES61 to the BCG strain, gaining the power to escape destruction and develop the disease. So great, now we have a target. We only need to solve the structure. The problem that was not exactly the easy answer. Well, this was actually just the beginning of a long and difficult journey. 20 years have passed since ES61 became relevant for the virulence of Mycobacterium tuberculosis. And although we have achieved some milestones like this, the structure of the inner membrane complex that you can see here, we still don't have the whole picture. We ignore which proteins form the outer membrane complex, limiting the possibility of solving a structure. And as this is the region of the secretion system exposed to the environment, we cannot design drugs or antibodies that can target it without it. There is some evidence indicating that protein substrates like the ones depicted here are proteins that are secreted to build this part of the complex. One of these proteins is SPB, which is the main project of my PhD. When I started working 6 years ago, we only knew that ESPB would be secreted through the machinery and that there was a protease called MICP that would remove part of the protein, and that after secretion, ESPB would make something bigger, a cylinder like made of 7 copies of the protein. But all this information was scattered in the literature and we could not make a story out of it. What we found is that the oligomerization of ESPB is actually triggered by the removal of the tail in an acidic environment. This was interesting because this is the kind of environment that you can find in the phagosome of immune cells. Later we also found that this behavior only happens in slow-growing Mycobacteria species, which are known to be pathogenic. So that made us think that maybe, just maybe, the oligomerization of the SPB had a relationship with the pathogenesis of Mycobacteria. But this is an open question that needs to be addressed. While working with the electron microscope, we notice a few peculiar things. One, and the most obvious, is that ESPB has a preferential orientation. What you're looking at here is only one side of the protein. This is a common problem in cryo-EM, although not as strong as this one. And what happens is that some part of protein interact with the air-water interface, which is a hydrophobic surface. We found out which protein region is causing this problem, and visual inspection confirmed the presence of hydrophobic amino acids, which made us think, together with some other information, that this behavior could be mimicking something that also happens in real life, that ESPB interacts with either the outer membrane of Mycobacteria or the phagosomal membrane. The second curious thing was the presence of dots inside the protein, as you can see here. After processing the data, we found one ESPB inside the cylinder. confirming that ESBB could allow the transit of other proteins through its pore, and supporting the idea that ESBB could be one of the outer member components. How exactly and where its position is still to be investigated. In all secretion systems, you will find proteins that work as chaperones, which assist the secretion of other proteins. In this particular system, the most studied chaperone is ESPG, which brings the BBB proteins to the secretion system. We were not sure if ESBB needed a chaperone, but there was some indication that if any, it would be the protein ESPK. So we investigated if that was the case. And by using a mixture of techniques, we would describe the structure of ESPK with ESBB. And to our surprise, the interaction is almost identical to the PBB together with its chaperone ESPG. That was unexpected because ESPK doesn't look like ESPG at all. But this supports the idea that ESPK acts as chaperone for ESBB. Not sure if it brings ESBB to machinery, so this model is still very speculative. But we discovered that ESPK stops ESBB from forming the oligomer. Keeping it in a single unit allows the protein to go through the pore of ES61. Otherwise, it would not fit. This, of course, opens up some interesting possibilities. If ESBB is so important for ES61, and the oligomer form of ESBB is a functional form, and if we have a way to break the oligomer, like let's say a molecule that looks like ESPK, then maybe, just maybe, we can stop ES61 from functioning, and thus the bacteria developing the disease. I know. There are a lot of ifs in that sentence, but we need to start somewhere, right? And I genuinely believe that ESBB could be an excellent target for new drugs or vaccines against tuberculosis. So in summary, what we did was to solve the structures of elements of the ES61 secretion system for Micromaterial tuberculosis, aiming to understand their function. This allows, hopefully soon, the design of vaccines that will protect us from getting ill, or new drugs that will be effective and non-toxic for our body. And that is actually our new project, so I'm very excited about it. This is, of course, just a very, very quick summary of some of my work, not even everything. But there is unfortunately not time to go through every detail. So I hope that at least this gives you a good idea of what I did. Thank you so much, and I'll give you the word back to the prorector.
Thank you very much, Ms. Candidate, and also thank you very much that you stayed in time. You just used 12 minutes, which means there will be 3 additional minutes possibility for the members of the opposition to ask a question to you. Very nice. The opposition will be then opened, and the opposition will be opened, of course, by the chairman of your assessment committee, Professor Reutlingsberger. He is the chair in biochemistry of apoptosis at Maastricht University, and I would like to give him the first opportunity to ask questions to you, Professor Reutlingsberger.
Unmute.
Yes, you are.
Can you hear me?
Yes, we hear you.
Thank you, Mr. Pro-Rector. Dear candidate, I would like to start with giving you my compliments for your thesis, your work, and also the very clear introduction, the presentation you have given. And I'm really impressed by the work. I read it with much pleasure and extremely much interest. And I was really also impressed by your general discussion chapter where you ventilated your societal engagement, which I don't see that often in thesis. So I liked it very much. I was very much impressed. My compliments go also to the promotion team. Without that team, I think you could not have achieved what you have written down in your thesis. So also their compliments. And I would like to start my discussion with you about something in the general introduction. And there you write that, of course, in the light of your philosophy, that the structure of proteins is the basis for understanding and for strategies, novel routes towards therapeutic interventions with drugs. And you write something which I find very interesting, and I think it's true that the protein structures elucidated by crystallography is a snapshot of the functional cycle of a protein. So you make a snapshot of the structure. You also say that the proteins have long disordered regions, and some of which you call primarily unstructured. What do you exactly mean with primarily unstructured regions? Can you explain that?
Well, all that section, what I meant was that originally we always thought that that proteins were fully structured, and that was the most important part of the protein, the structural part. But what now we know is that actually not, that there's a lot of parts that are flexible, some really big parts that are, well, are considered completely unstructured or partially unstructured. And those are the most interesting parts because those are the ones that can move and they can do things. Yeah. So, and for example, I mean, there are many proteins that are, the fact that they are or have these big regions that they're not structured, that can do their job. I mean, I think a nice example is the, I believe the nucleopore that has so many, that the only thing they need to do is like hair that are just pushing the proteins through it and so on. So yeah, I mean, I don't know if I answered my question. But
yeah, so it was not to indicate that that in time, most part of the time that region is unstructured, but there are also parts of time that the region is structured.
Yeah.
And you can pick up with your crystallography, you can pick up the structured parts, because you need structured parts to inspect, and you can visualize them with all kind of analysis. So I was in the direction that there are parts that are primarily unstructured, but there are also times where these parts are structured, and you can catch them. You can so then you have to do many analysis in order to catch the particular structured time point. But that's is that the understanding correct?
Well, I think the thing is, and I think that one of the beauty of proteins are that there are just so many that basically you have almost an example of everything, which makes it difficult, but at the same time very interesting, of course. Like you never stop working with them. I mean, there is people that work their entire career with a single protein because there's just not enough time to study it, everything.
Yeah.
And so yeah, sometimes, and something that I also wrote down as a quote from Heisenberg, which I loved it when I found it, was that we have to remember that what we observed in nature is not nature itself, but nature exposed to our methods of questioning. And it's so true, because it really depends on what we're asking and how we're asking it, the answer that we're going to get back. And it's not necessarily the full truth, right?
Mm-hmm.
So
Yeah.
sometimes you'll have proteins that are normally unstructured, and in a very particular case, it will get structure, or maybe it will stay unstructured because that's the way it needs to be. That's just.
I would like to discuss this with you for a long time because I'm very much interested. But considering the time, I would like to switch to the ESPB protein, which was the main subject of your investigation, where the C-terminal domain is unstructured.
Mm-hmm.
And while you were doing the work on that, revealing that structure, and you've written that down in chapter 4, there was this group with Python that published the structure of the SPV using the same technique, using the snapshot or the single particle analysis with the cryo-EM. You cited the study, but you didn't discuss the results in relation to your results. At least not what I would have expected. And so I would like to discuss that with you and ask you, there is one, what I found significant difference between both structures published, and that is the pore size of the heptameric complex. Both you have a resolution of around 3.4 angstrom, and you come up with a pore size of 40 angstrom. So that's the gap in the heptameric complex. And they come up with a pore size of 45 angstrom. Now, is that difference significant to your opinion? And can you explain, or have you an idea why there is such a difference between both structures?
Well, 5 angstroms.
Yeah, 5. With a resolution of 3.4. Is it relevant?
Yeah, that's so one of the reasons that I didn't talk too much about that structure is that I guess unfortunately we notice many, like, pretty much the same thing.
Yeah, that's true.
We reach a higher resolution, which we were really proud of it. But there was a shame that it was not strikingly different, which of course that's a shame, because we were late. If the pore difference is significant, well, I guess I'm not entirely sure how they come up with that number. I literally measured even in Chimera, like from one edge to the next, and even I use another software called Pore, which gives more or less that number. So I'm not entirely sure how they measured, if that was accurate, of just roughly.
So both of you made use of recombinant proteins expressed in a bacterial system. And I think both of you observed that if the C-terminal domain is present, you get a preferential localization of your particle on the grid.
Yeah.
And you have chopped up put away the C-terminal domain, and then you saw a random orientation of the particle on the grid. And they used the fluorinated octyl maltoside, which is a detergent. So they looked at the whole protein with the C-terminal region attached to it. And maybe that explains the difference. I don't know. I was hoping that you could give me that answer, but from let's say that this
Professor Reuteningsberger,
Yes.
Sorry to interrupt, but can you phrase your question shortly? And I also expect a short answer back from the candidate. Thank you very much.
Okay, very short is if you had to choose a structure for structure-based drug discovery, which structure would you choose? Yours or Python's?
Well, I truly believe that in this case, the functional part is the structure part. So for me, the difference between their construct and our construct wouldn't make a big difference in terms for drug design.
Okay, that's a good answer.
I think you have to close your.
Let's keep it like that.
I'm very satisfied with your answers. Thank you very much, and I'll give the word back to the prorector.
Thank you very much, because due to time, and we are really 2 minutes behind time, I would like to speed up, and I'm going to give the word to the next opponent, which is Professor Sixma. She is also a member of your assessment committee and affiliated with the Dutch Cancer Institute in Amsterdam, and the head of the Department of Oncology, Biophysics and Biochemistry, and I'm gladly like to give her the word. Professor.
Thank you, Mr. Prorector. And dear candidate, I would also like to congratulate you on a very interesting thesis. I enjoyed reading it. I don't know all that much about microbiota, so I learned a lot. And I enjoyed also the many different structural techniques that you have used throughout your work and nice combinations of that. I would like to have some questions when I looked at it, and that was especially on chapter 6, where you study the SK protein and its complex with SB. And you use different techniques, and you also do use AlphaFold. But what I wanted to ask you is, we are also happy with these new opportunities with the AI, expanding our structural arsenal. We're also happy with the opportunities in cryo-EM. So I think altogether, we have many opportunities now that were not there previously. What I wondered is, did you get a chance to look at the full-length structure prediction for SK? And what did you think about it?
Yes, we did. Of course, the moment AlphaFold or that paper came out, or in that browser came out, like immediately we started Googling, or getting the structure of all our proteins. Yes, and it is actually in general for all the proteins from this secretion system, they're very peculiar in the sense that it is like a structured part and like a rope around it, completely unstructured. And that's what we already kind of knew for ESPK. Our experimental data luckily supports that idea. Well, not luckily, I mean, I guess it's a nice confirmation.
Okay.
Yeah, so we tried before also with ITATR and unfortunately ITATR couldn't handle the whole protein. So we need to split in several parts. But even combining it all seems to have the same.
Yeah, so I was more struck by the fact that the first 180 residues are actually folders in AlphaFold. I don't know if you remember that now.
Mm-hmm. Yeah.
And that position, AlphaFold positions that, to my surprise, in the place where your SB is bound, if I'm correct.
Can you repeat the?
So the first 180 residues of SK are folded.
Yes.
And maybe I ask because I have many questions about this. I find it very interesting. It is a helical bundle.
Yeah.
To my uninitiated eye, that looked very much like your SB.
Yes.
And when I superimposed it, it still looked more or less similar. So one question I had there, this SB is one of the PPE-PP proteins. So
Yes.
was it expected that this N-terminal domain would have this same structure? Is it a member of that subfamily of PPE-PP proteins?
Okay, I see that now. Yes.
Two separate questions, really.
Yeah, but I roughly understand what you mean now. So yes, it is this two helical bundle, or sometimes more. It is very common. They not necessarily are from the same family. So the PPPs, for example, they're called like that because of the motif, the PE and PPE motif. ESPB have those motifs. Well, no, but they belong to the same family. But in this case, ESPB is not. It does look like, and actually when, at least, ESPK, sorry, at least when I was playing with ITASR, by our surprise, the proteins that were used to build that model were PPPs. It doesn't have the motif, so I guess it's not, it wouldn't be considered a PPE protein. But you mentioned that it's placed in the same binding to the...
So roughly in that same area. So now that I saw that, it looks like ESPB, it sits in more or less the same space. What is it possible? Have you considered that the full-length protein, this would actually sit there? It's not a strong interaction in that AlphaFold prediction, but you know, it's quite striking that it sits there.
Yeah, that's something that it's still, of course, we need to test. When we were doing, for example, circular dichroism, we got the melting temperatures, and by our surprise, the full-length ESPK was less stable or had a lower melting temperature than the C-terminal alone. So our reasoning was that if the two domains will be interacting with each other, maybe should be more stable, not less stable. But that, of course, is not the right technique to prove that. So one thing that we were always curious and wanted to do, unfortunately, there was no time for that, was to produce the N-terminal of ESPK by itself and test it. But unfortunately, we don't know yet.
Okay, that's good. So you were able to make the full length because that isn't explicitly in your thesis. Mm-hmm. And I was wondering, you've shown that your ESPK C-terminal domain can disrupt the multimer of the ESPB. Can the full length do that?
Huh. We never try that.
Yeah, because if it's already occupied, maybe it doesn't do that.
Yeah, that's a good question. We never try that, and a very naive answer is that we didn't like the full length. It's a complicated protein to work with, but no, that it's a shame. But no, we never try that.
It's a
sorry, you know that one of the reasons that we didn't try that, and is because of the shape of the SPK. So we, because, well, the instruments that we had in the lab, we use a lot of gel filtration, and an ESPK full length in gel filtration makes a very nasty profile because of a whole flexible part, and it's gives a lot of peaks and so on. So it was very difficult to distinguish between ESPB alone, the complex, and the protein. We should have done that with EM, of course.
Would be interesting, I think, to do that. Do I still have?
There is time for a short question, yes, and a short answer as well.
Yeah, because I'm still to go on with your what was it, circular dichroism result.
Mm-hmm.
Do
Yeah.
you think the protein that you made there, this full-length protein, is in the state it should be, or is that just a poorly made protein, given also your gel filtration results? And where was that made? And are there other systems to get? Yeah, I know some of our proteins, they're terrible if we make them in E. coli. They're great if we make them in insect cells. In this case, I don't know where you made this protein, and would that be a possible way to get around your problem?
A short answer, please. If not, we
Sure.
will proceed. I'm sorry.
No, I think I
Okay.
can quickly answer, or at least sort of. We did in E. coli, and of course, there are several examples that making it in Microbacteria makes a huge difference. So the quick answer is not sure exactly what if it would make a difference. I think it was definitely worth to try.
I'm very happy with that answer, and I happily give the word back to the project.
Thank you very much, and I'm gladly can we proceed. The opposition will be continued by Professor De Jonge from Antwerp University, from the Department of Microbacteriology. She was also a member of your assessment committee, and I would like to thank her that she joins all the way from Antwerp on the digital way to this thesis defense. I'd like to give you the word.
Thank you, Mr. Prorector. And thank you, Abril. I learned a lot, especially on cryo-EM and all the possibilities from reading your thesis. So in chapter 4, so you also show in your nice presentation that you just gave on slide 11, the model 1 and 2. And of the 3 models, which one do you think to best approximate the truth, and why?
It's definitely not an easy answer to give, or a short one. The thing is,
You probably have a favorite. Which one is it?
My favorite is the last one, just because it's a little bit more crazy or unexpected, and I'm still. It isn't for. We have some evidence that ESBB makes something bigger than heptamers. And we thought about this so much. Unfortunately, we haven't been able to find a condition to test it, or like to make it stable and study it. That is definitely unfortunate, because I think if that would be a great piece of structure for the outer membrane. But even if that one is not exactly true, and I'm just seeing what I want to see, if I need to, let's put it this way, if I need to put them in order of my favorites, I would go for number 3, so making this tubular form, the number 2 that binds to the phagosomal membrane, and then number 1 somewhere in the outer membrane of Microbacteria.
Thank you. And if you would be asked to select a target for an mRNA vaccine for TB, do you think ESBB would be a good one, and why? And which domain?
Yes, I really think. I think ESBB should be exposed enough to be a target. mRNA sounds a very nice way to go. Of course, it seems we have proved that it's working, and it's definitely a good option to go. Which domain? Well, how to construct that vaccine? I would go for one of the constructs that I have that it's the most stable, which is ones that is cleaved after MIG-P. That one oligomerizes the best, and I think we definitely need to keep in mind that it needs to be an oligomer to target for the vaccine to target it. Yeah.
And if you would say, you also explained on the Tundra EM, and the bottlenecks of performing cryo-EM access in low and middle income countries. So if you were handed a Tundra EM and unlimited amount of money for consumables, so what questions would you address next? And would you be able to resolve model 1, 2, or 3? And how?
Those are, I think I wouldn't give, I mean, my supervisor are going to kill me because my answer is not EM actually. Well, not at least with Tundra. And the reason is Tundra right now, unfortunately, is just for a single particle. And I think I will be more interesting to go for tomography. I have many experiments already in my mind, and I was already like working on them when the corona hit and everything kind of slowed down or completely stopped, to prove one model or the other. Which actually, it was not even with EM, it was just with fluorescence. But unfortunately, that we couldn't continue with that. But with a microscope, the experiments that I would do that I think will give the answer is with tomography, trying to visualize the outer membrane or the whole machinery together. We have, well, not myself, but the whole community are trying to find out how is built the outer membrane complex, what is making this part of, and no one has been able. I don't know anyone that has like a solid clue or solid evidence. I mean, I would consider our results the closest to something, which they're still not conclusive. So no, I think not even purification, it's the like pure biochemistry or classic biochemistry will give us something. I think we need to go beyond the classics and go for more state-of-the-art EM, which in this case is in situ cryo-electron tomography.
And regardless of the technique, if you indeed want to detect something sticking out of mycobacteria and not look at purified protein, it's not easy to find proof of something not happening. Huh? And if it's a rare event, how do you balance the approach of screening large surfaces of mycobacteria for a possible signal, which you then confirm? Would that be with fluorescence, or how would you go about that, or this tomography?
Yeah, that's of course what has stopping us from getting anything. Well, I guess that's not nice to say, but that's the case. I mean, we have tried really, really hard, and unfortunately, we have tons of data, and still we haven't been able to see. Oh, this is ESX1, you know. Of course, fluorescence, so CLEM, correlative light electron microscopy, is a very popular combined technique to find, well, the needle in the haystack, of course. That's not straightforward. Something that we were working on, it was to make a mega tag. This idea came from my colleague Axel Sirois, and it was to use a super big protein, in this case encapsulin, and engineer it in a way that it can just stick to the complex and see it easily. I truly believe that is a very good method or interesting tool for tomography. We normally also use labeling, but that also it's there. it's not straightforward, unfortunately. I would, if you ask me which one I would choose, I think it's worth developing the megatab of encapsulating as a tool to find out proteins inside the cell.
Thank you. I'm satisfied with your answers, and I hand the word back to Prorector.
Thank you very much for your questions. And the opposition will now be continued by Dr. Nicolas. Dr. Nicolas was also a member of your assessment committee. He is affiliated with the Department of Biochemistry here at Maastricht University with a specialty in molecular biology. And I'd like to give him the word. Dr. Nicolas.
Thank you, Mr. Prorector and dear candidates. Also, I would like to thank you for the very clear introduction that you have given on this interesting topic. The thesis was well written and it describes research that is very important and very timely. And you have used techniques here that combine solid biochemical and molecular biology with state-of-the-art structural work. And this is really a thesis to my heart, I would say. It's given me excellent insights in this fascinating field of structural experimental biology. And you and your supervisors did an excellent work. And I would like to congratulate also your promotion team. Together you have made a really good contribution to the field. And with that, I would like to move on to the questions that I have, because I enjoyed it very much. And already in the introductory chapter, you came up with an interesting analysis, because there you presented that there is an effect actually of the current SARS-CoV-2 epidemic on the apparent increase in tuberculosis cases when you view this globally. And as it happened, just a couple of weeks earlier, I read a paper on quite an opposite effect, where they saw that there was a drastic decrease in the cases of influenza also on the general population. So here we have a pandemic that is causing two very opposing effects, one side on the tuberculosis, but on the other side on influenza cases. Can you help me set this straight, or how should I see this? Because I would say in both cases, you need people to meet.
Yeah. That remind me of when everything, we started with the pandemic. Even like I remember the day, because Bauke came to your lab to give a lecture and the pandemic was just hitting. So the reason, I mean, there is evidence that tuberculosis unfortunately has been affected. The reason is that, I'm sorry if there's some background noise here. So the reason is that I would say, okay, so the situation now is that you're supposed to be at home. There's not much contact with other people. You have, in theory, face mask somewhere sometimes. And that should like stop spreading bugs or sharing bugs between each other, right? Yeah. Unfortunately for tuberculosis, I think the question is it stopping from spreading, or is it stopping from getting treated? Like the question is tuberculosis coming in, or the problem is that it's already in and it's not being treated? So that was the number of cases is, and I think Bauke will correct me if I'm wrong, those are still estimates. Unfortunately, hard numbers, well, I guess also with this pandemic, they're still estimates. We don't have like accurate numbers. But still, the number of cases went down. And it was not because there was less people getting ill. It was just because people were not going to the doctor to be treated and to get tested. The number of deaths I believe, I think, oh, I wasn't sure. Now I'm
The
not
exact
sure.
numbers are not so important here, I think, in the discussion, but the comparison to the influenza is what I believe is an interesting one. Do you see
Yeah,
differences in the mode of transmission?
well, I think the way of transmitting is very similar. So why there's such a difference, not entirely sure. I think in the case of tuberculosis, the main problem is that people are just staying in their home without treatment, or the treatment cannot reach them for, well, resources are being moved, or just not enough people, and so on.
Okay, I think it's the answer so far is already has shown that you have thought about this, and probably a lot more can be said about this. But I would like to move on to another question, if you're okay with that, and that's about the chapter 3, because there you describe how you sort of want to use ferritin as a gold standard for cryo-EM, and that the current preparations of this protein, they do not allow a real comparison between machines or between different laboratories. That is how I understand. And my first question is, why did you choose for ferritin, and what in general would be the ideal protein to allow for such standardization?
Well, ferritin, it's a beauty for cryo in the sense that because it has a very nice size, so quickly in the microscope you can see the protein beautifully. It's symmetrical, so you don't need too many particles to have a full reconstruction. So those are like the two main things, and it's very soluble, and it's, well, I guess, yeah. So you can have the protein. It's easy to work with, and it has a great size for a cryo to see it. It has symmetry, so you don't need too many particles to quickly get a full structure. Because of course.
Does this also mean that ferritin then that holds all the peculiarities that other proteins can have? And would it not be more ideal to have sort of a standard set of proteins to test your machine? Or would it be the same for all of those special proteins?
No, of course. I mean, there are a few, for example, beta-lactamidase is one or another protein that it's very commonly used to test your machine, which is very different. It's smaller and it doesn't have the same symmetry. And that, of course, comes with some other, well, you need more particles, you need more micrographs, it might take some more time. But of course, I guess it depends on what you want to test. So for example, when we were developing the Vitrojet, we needed to test many different proteins because we wanted to prove to the community that they can use the machine for everything. So in this case, to test your microscope, just to test if your microscope is running fine, I would go just for ferritin because of how easy it is to visualize and to get the structure. If you want to test something else, I will go for other standard proteins. The problem is that there are not many standard proteins that are so accessible that you can maybe purchase in Sigma or something.
Okay.
That are affordable.
Thank you. I'm looking at the prorector. Is there still time for a short question?
I'm sorry to have to disappoint you in this matter.
Okay, then I'm very happy with the answers you've given. I'm giving the word back to you.
Thank you.
Thank you very much, and thank you for your questions. Well, we're going to continue with the opposition with your final opponent, and I would like to give the word to Professor Smeets. He is affiliated with the Department of Clinical Genomics at our university, was also a member of your assessment committee, and I'm gladly give him the final word for opposition.
Thank you, Mr. Prorector. Dear candidate, it was a pleasure to read your thesis. Compliments to you and your promotion team, not only because of its scientific quality, but also because of the strong position you take in arguing that TB is not only caused by bacteria, but also by poverty. I fully agree on that. Therefore, it struck me that you referred to Bill Gates to support your point. You mentioned that Bill Gates gave a TED talk in 2050 where he suggested having an army of healthy workers with the same system as the military, paid people that train every day while waiting for their mission to save lives in their own country or elsewhere. You indicate that he has a point. This pandemic exposed how unprepared we are to deal with wars against microscopic enemies. We need governments to invest in science and our medical system. But in my opinion, that will not take away the poverty as a cause, especially because the 10 richest men, including Bill Gates, doubled their income during the pandemic. I would not have said, Bill Gates, you have a point. I would have said, Bill Gates, you should be ashamed, as your wealth results in direct harm to the vast majority of ordinary people across the world. Do you really believe in people like Bill Gates to get a better society and an end to poverty-related disease?
No.
That's a good answer.
No, I mean, no, definitely not. I said that he has a point because I think it is true what he's saying. Unfortunately, I think we should have resources to fight these microorganisms. We should invest way more money than we're investing in health, in science.
Mm-hmm. But is it not the money is not the problem, but the distribution of the money is the problem? And Bill Gates exemplifies how wrong that can be.
Yes.
But I will not trouble you with these dilemmas. I will go back to the scientific part. So in your introduction, you discussed the life cycle of the microbe. And you said it ends in necrosis over apoptosis for bacterial content release. That's a bit of an old view, I think. I think apoptosis is being seen these days also as a way of distributing the disease and to distribute the infection. So I was wondering what your current opinion is on that, because that reference was on the year 2000. And if I look at apoptosis, I see one of the proteins is ESBC is quite crucial in initiating apoptosis and has probably a clear role also in this process. And being a mitochondrial fanatic, so I'm always triggered by things associated with mitochondria, but I was wondering, why did you choose for ASPB, whereas ASBC would be a very good candidate to look for a treatment or to look for a vaccine, because you know the downstream process, and you could, if you can intervene, then inhibit the spread of the microbe in the body.
Well, the reason is that so we in the lab, we were working with the type 7 secretion system, so ES61, and immediately, me as a protein biochemist, I was really interesting to work with this protein. I thought it was fascinating. I didn't know the other protein, so that's a simple answer, is that I was just I like this one, but that's not a good answer, of course. I didn't know enough to really choose between one and the other. To be honest, my previous background, it was in ribosome biogenesis, so, and in yeast, you know, like, so yeah. Micromaterial world was completely new to me. So unfortunately, I didn't have.
Yeah, but now in 2022, what protein would you then choose now, with all the knowledge you gained in this system, to be the one which you would like to address to reach the point you want to reach, because your ambition is quite clear?
Yeah, but you're asking me now that I'm in love with the SPV.
Sorry?
So you're asking me now that I'm in love with the SPV. I've been
with the whole ESX1 system. Yeah, you say they produce multiple, they secrete multiple proteins. Which one would you then like the best? I think you've shown that you can gain way big progress in your efforts, but if you would now evaluate what I would choose my next candidate as, so that might be a better question. And why would you then go for that one in developing the vaccine or in developing ways of combating the disease?
Yeah. Well, the thing is, I'm not yet done with the SPV, so I'm still will be working with the SPV. I have so many questions, and I think they're really important to understand the SPV in order to understand the ESX1.
Sure. But
So,
you claim you're looking for a vaccine or treatment, so then it's not only what interests you, but also what would the protein yield that.
No. But I'm still genuinely believing that ESPB it's a great target.
Yeah.
So for example, ESPC, of course, it's interesting. It's known to make this filament. At least there is some evidence of that.
Yeah.
Unfortunately, that group didn't go forward, or at least they haven't been published anything about it.
Yeah, I think it triggers a stress response. So it's an ER unfolded protein response, which then indeed triggers the apoptosis field. But indeed, blocking apoptosis could help spread downstream effect. Whereas for SPPB, I still think you're troubled what it really does in the end, isn't it?
Yes, of course. I mean, that function is still to be unraveled. But I don't know what to say.
Oh, sure, you're in the defense now.
No.
Well, you heard ora est, which means this is the end of your defense. And I would like to mention that we have to stop now the questioning of the last opponent and the answering of the candidate. Miss Abril Gijsbers Alejandre, Miss Candidate, the time appointed for defending your thesis has now passed. The degree committee will withdraw online to discuss not so much the quality of your thesis, because that has already been approved, but we will discuss the quality, the way you have defended your thesis this afternoon, or for you this morning. I request that you and your online committee, your online family, will wait till the results of our deliberation and our return in the next 10 to 12 minutes. Thank you very much.
Thank you.
So. Iedereen is er? Yes, okay. Dear Ms. Abril Gijsbers Alejandre, the degree committee here present online has assessed the quality of your thesis and the way you have defended your thesis for you this morning. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor, and I can see your big smile, so the body language is also online visible. Professor Peters is authorized to confer upon you this academic distinction in accordance to Dutch university customs, and therefore I invite your supervisor now to take the floor and use all the official statements. Professor Peters.
Thank you very much, Pro-Rector. Dear candidate, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Abril Gijsbers Alejandre, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor, affixed with the official seal of the university shown by the beetle.
Thank you so much.
So, is this official part now being completed, dear candidate, dear Dr. Gijsbers, dear Abril? We have time for a laudatio, and the laudatio is divided up in two small parts, one by me and one by my colleague Remo Ravelli. But on behalf of Remo Ravelli, but also the other co-promoter, Carmen Lopez Iglesias, we congratulate you with this enormous, great accomplishment. You were the first coming as a PhD candidate to our laboratory when we were setting up the institute from scratch, and you worked very hard for many years and built up with us a team. You are really a pioneer. Remo will go into the details, but I like to highlight some of the skills that you have. You have an enormous amount of transferable skills, very important for your future. You earn trust and confidence. You convey a vision. You drive a point home, and you're very good in persuading. You are very good in overcoming controversies, and you are an expert in motivating others to action. And we have seen that by the senior staff, but also by all the students that you've been taking care of. You were able to convince students to the project that they're working on is important, convince students that they are important, and convince students that their contribution to the project is important. You have the quality of winning hearts and minds. In the lab, we could see passion, risk-taking, outward-looking, flexibility, stamina. You were very result-oriented, independent, and you showed creativity. In the lecture hall, you were able to grab attention, explain complex ideas in simple terms, and you were really good in increasing your own visibility in the lab and also abroad. And besides that, setting up collaborations is also a very important characteristic of you. You looked for sparring partners to develop your ideas, step over the borders of your discipline, Communicate your wishes, doubts, and fear. Use feedback to your advantage. And you had intellectual initiative. You have a critical reflection, the ability to identify and solve problems independently, and interacting successfully on a one-to-one basis. You have really very good observational skills, and you are really a people's manager. And then in the last half year online, we saw also the quality of your PhD thesis writing from Mexico. The ability to present and write material in a very clear and appropriate way, and performing the text completely on your own of the introduction and the discussion with your own, let's say, approach. And today you heard how much that was appreciated. So on behalf of Remora Valley, Carmen Lopez, and also on behalf of the candidate, I like to thank the Assessment and Defense Committee, as well as the Pro-Rector for their great and critical work and the valuable time. And I'd like to give the word now to my colleague, Ronald Remora Valley.
Dear Dr. Gijsbers, dear Abril, I would also like to congratulate you with the successful defense of your thesis. It's the end of a long journey which started in April 2016. You directly impressed all of us with the superb elevator pitch at the link opening. You demonstrated passion, drive, dedication, and a mission, which you continued to do so through your entire PhD career with you. You have been an inspiration, not only for your colleagues, but also for your collaborators and the many students you supervised. You helped us setting up new courses and taught us as tutors. One of the students wrote in their evaluation, Abril was the best tutor. She is amazing, and she really spent so much time helping us and making sure that we had good feedback. She made the course so much fun. Abril, you have been our very best first student. That's a lame joke, as you are the only, you are the first student to defend your PhD. Previously, you expressed that you tend to wave away any compliments if they don't come together with content, and there's ample of content. Let me give you some examples. You have been the first in our lab to bring a project from the start till the end, from structure to function. You have been the very first natural Navy NASEM user. You have solved 3 SPA structures at better than 2.3 angstrom resolution. That was a seminal resolution when you started your PhD. You won the NVK Best Presentation Prize. You are the first and only one, and possibly even the last one in our lab, who combined so many biophysical characterization techniques in one booklet. You were the first to embrace writing retreats. You had brought our group meetings into a structure it badly needed. You've been the only Dutch person in our lab who doesn't like to cycle or doesn't speak Dutch. You have been babysitting even in the middle of the night. These included real babies, as well as your biochemical lab experiments. You thought of proteins and structures miles beyond our first observations as a professional chess player who could think 10 steps ahead. Dear Abril, a big congratulations with this important step you made today, and all the love and best for your next steps you have in front of you, together with your loved ones in Mexico. I also like to take some time to congratulate and to thank your loved ones. First of all, your mother. You allowed and supported Abril to move so far away in times that must have been difficult for you. Abril could go either to Cambridge or to the Netherlands, the county which you have met your husband. Abril always talked very warmly about you. One of the things she told me is that she was raised as an atheist. She assumed that you were both a-religious as well. And she was surprised to hear her father saying during a car trip that that assumption might not have been necessarily correct. I would believe that, wherever he is now, he would be very proud of you as well. Ik zou graag opa in het Nederlands toespreken. Meneer Gijsbert, van hartelijk gefeliciteerd met het behalen van de dochterstitel van uw kleindochter. Wat fijn dat we u hebben hier mogen ontvangen in het lab in Maastricht toen ze net begon. En wat fijn dat Abril zo vaak thuis bij u kon komen om spelletjes te spelen en oude fotoboeken te bekijken. Zij mist deze bezoeken heel erg en wij missen de verhalen over u. Maar vandaag is het feest. Uw kleindochter is met vlag en wimpel geslaagd en gepromoveerd. Dr. Gijsbert, van harte. Joanna has also been in our lab. I didn't know she was there. I wasn't introduced, saw her from your back, and almost mixed the two of you up. But Abril explained me all the differences. Joanna didn't want to stay just in the Netherlands, but explored more exotic places on this earth. You bought a home base together in the city I grew up with, Utrecht. World travelers, but still close together. A big thanks and congratulations to your sister as well. Just before I started my own PhD, I worked a few months in the very south of Italy together with Dritan. He had just left his home country and started his academic career in Bari. How miraculous life can be. Dritan stayed in Bari, built up an international network, and started collaborating with Nuria. The posters on the wall I saw during my last visit in your lab still witness a fruitful collaboration. Gijsbert, Dritan's liquid, and Sanchez Buick. He spoke highly about this great biochemist from Mexico, who was not only a good scientist, but also a good teacher. Thank you so much, Dritan, for having brought Maastricht on the attention of Abril and Nuria. Nuria, also big congratulations to you. I would believe that Abril possesses so many intrinsic factors that makes her such a special person and a good scientist. But you nurtured them all and you let them grow and flourish. Your training and support was not only evident when she started with us, but always there in the background throughout the PhD. It gave a fruitful and healthy reflection on most occasions, albeit I must admit, it also gave me 1 or 2 sleepless nights. I especially also like to thank you for the last 10 months back in Mexico, in which Abril could finish her thesis, resulting in our festive day we have today. Big congratulations and all our love and best for your future. With that, I come to an end of my congratulations and the end of the laudation. I would like to give the word back to the pro-rector.
Okay, one moment. Well, thank you very much, but I was already informed this is not the final end. There's another word, and I think also the third promoter would like to address shortly the candidate. So I would like to give also the word to Ms. Dr. Lopez Iglesias. Would you like to address briefly the candidate as well?
Yes, thank you very much, Pro-Rector. So I would like to congratulate the candidate, Dr. Gijsbers, now, but mainly I would like to address some words to the family and friends in Mexico. Quiero agradeceros a todos vosotros, querida familia, queridos amigos, haber tenido la oportunidad de tener a Abril con nosotros y haber disfrutado de su formación, que es vuestra responsabilidad totalmente, y su manera de ser, su educación, todo lo que ella ha contribuido a nuestro grupo. Y quiero agradecer a su madre, por supuesto a su padre, y a Nuria y a todos sus amigos. Sin vuestro soporte, vuestro apoyo, a Abril esto nunca hubiera sido posible. Gracias, Abril, por compartir tantos momentos con nosotros y por ser tan bella y linda persona. Gracias a toda la familia y a tus amigos. Un beso enorme. Thank you very much, Pro-Rector. I finished already my words.
Thank you very much. So I'm going to also would like to address the candidate now personally myself too. So not Miss, I would say officially now, Dr. Gijsbers, I would like to congratulate you on reaching this point. This is a very special moment in your life. You have shined today. You did an excellent defense and you get this reward afterwards. In my congratulations, I also would like to congratulate the promotion team, the 3 members who over the last 6 years has worked to you, made some very nice publications together and really access this moment and this booklet which you produced today. I further also would like to thank the members of the Corona who joined us today, took the time to prepare. Sometimes I'm saying that it just takes 10 minutes questioning, but it takes much more time to read the thesis and to make the questions and to get the answers. And again, I would like also our special thank to our guests from Antwerp and Amsterdam that they would like to join us. My congratulations, of course, is going also to your family who is online, who we unfortunately cannot see in Mexico. From my personal perspective, I studied also myself for 1 year in Mexico. When I was a student, I was in Querétaro, which is just north of Mexico City, and I know the place quite well. And I think you did a great job. So officially saying to all the members which we are now in Mexico, the family who have watched this event, that you did a great job. And I'm going to close officially the ceremony. And as a pro-rector, I have to use my hammer, which is right here. And you can hear here that I use my hammer and I close it officially, this ceremony. Thank you very much. And this is over.